Aplysia californica is a marine mollusk that has been used for the study of the neural basis of behavior. Today, I'd like to describe the way in which you can use Aplysia to understand how you can use the same muscles and the same motor controller to generate a whole variety of qualitatively different behaviors. And this, I think, is a general principle for motor systems. It's not at all confined to how Aplysia behaves. The three behaviors we're going to look at are biting, which is an attempt to grasp food which fails, swallowing, which is a successful ingestion of food, and rejection, which is taking an edible material and pushing it out of the feeding apparatus, or the buccal cavity, as it's called. So we're going to go through that, and you will see that the different muscles and motor neurons that control these behaviors are deployed in different ways in order to generate them by the same animal, and sometimes within seconds of one another. So let's first look at a biting behavior. Here's a movie of the animals that attempts to bite, and I'm just going to draw your attention to the central structure here, which is coming out. This structure is known as the radula, and as you can see, it's protracted, and at the peak of its coming out or protracting, it closes, and then it goes back in. And in this circumstance, you can see the animal was eager to ingest the food that's here, but was unable to do so. How does that work at the level of the muscles and the motor neurons? This schematic is related to what you saw in the previous video. What you should imagine is that we've taken a cross-section right through the animal. Here's the esophagus, here's the line of the jaws, which is what we were looking at. And what we were looking at, we saw the top of this shaded region. This is the radular surface. And this whole schematic is based on magnetic resonance imaging recordings that we've taken. And these schematics allow you to see from within the animal how the different muscles and motor neurons are being activated to generate the different behaviors that we'll be talking about. The first stage is to push the grasper out, and that requires the contraction of a muscle behind the grasper, known as the I2 muscle, which is innervated by four neurons, B31 and B32, which are both motor neurons and interneurons, and B61, B62, which are motor neurons. And we can see this on the record here. This is recordings from an intact behaving animal in which we've implanted electrodes on muscles and on nerves. And on this muscle, the I2 muscle, we can actually see electromyographic recordings that show the activation of that muscle during the protraction phase. So we can actually see both the activity of the muscle and we can see the movements at the same time. Here, the next stage, as I pointed out to you, was that as the grasper protracts, it also closes. And that's due to the activation of the B8A, B8B motor neurons, which activate the I4 muscle, a horseshoe-shaped muscle, which causes the closing. That closing has two important consequences. As you can see, it changes the shape of the grasper from roughly spherical to roughly ellipsoidal. And in so doing, it pushes back on the I2 muscle, lengthening it. That has two very important effects. That increases the mechanical advantage of I2, making it stronger again for pushing the whole grasper out. It also puts it higher on its length tension curve, and both of those effects allow I2 to be, once again, a more effective protractor. You can see that as it shortens, if it shortens enough, it will no longer be able to protract at all. So by closing in the middle of protraction, the animal's effectively modulating the force. Now, the other thing to notice is that there's a structure here that's being stretched called the hinge. So let's look at the closing motor process, and that's the activation of the B8A, B8B motor neurons, which you can see here on the radular nerve. Again, this is a recording of the same intact behaving animal. And as I mentioned, you can also see that there's a structure here which we call the hinge. It's the interdigitation of the I2 and the I3 and the I4 muscles at the base here, and it's been stretched. And that means that now that the animal has failed to grasp, it can retract, and that mainly is done by activating the hinge, which is innervated by the B7 motor neuron. And so you can see activation of the B7 motor neuron. And as a sort of afterthought, there's also an activation of the jaw musculature, the I3 muscles, which are innervated by the B6, B9 motor neurons. You can see that's weakly activated here. But the main players are the I2 protractor muscle, the activation of the radial nerve in the middle to enhance I2's ability to protract, and then the hinge activation to retract the grasper. So that's our understanding of how biting works. Let's look at swallowing. What we're doing here is giving an animal access to a piece of inedible food, which is attached to a force transducer. That inedible food is double-sided tape with seaweed on either side of it, and you can see the animal's working hard to try to ingest it. Let's take a look at that process again one more time. You can see the animal releases, goes down, then it pulls it up so hard that it lifts its head, and then it again releases it. How does that swallowing of a heavy load work? Several steps are involved. First of all, once the grasper is going to be repositioned and has opened, the animal does not want to lose contact with the food, and so it pinches the anterior part of the I3 using the B38 motor neurons to do so. And that creates a period of time where it's actually pinching the food. And that activation of the B38 is shown here. As it continues to protract the grasper, however, it loses contact with the seaweed, and that's when you see the head move to the lowest position at the peak of the protraction phase, and that's due to the I2 muscle, which you can see is active here. And you can note that it's somewhat a shorter activation than in biting because you don't want to protract so much that you push the seaweed out. Now, the next step is that you need to close on the seaweed, and this is going to be the power stroke. You close by activating the B8A-B motor neurons, and then you're going to actually push the whole grasper back using the jaw musculature. So you activate the B8A-B here. You can see them on the radial nerve, and that's causing the closure of the grasper. And now the power stroke is the retraction phase where you activate the jaw, which is a piece of cartilage surrounded by the I1 and I3 muscles. The I3 are the very strong muscles that circumferentially surround it, and when they squeeze the whole lumen of the jaw down. They're innervated by B6, B9, and B3, and there is also some activation of the B7. And so now you see the whole picture. This is the B6, B9 here, the B3 motor neurons, and here you also see some B7 activation. There is some weak activation of the multi-action neuron we'll be talking about more before B5. So let's review the cycle briefly. Here you notice that the force has not reached its lowest. This shoulder is due to the pinching of the anterior jaws to keep hold of the seaweed. As the protraction proceeds, the animal loses contact with the seaweed, and thus the force drops to its lowest value. Then the animal both closes, as you can see, that's where the force starts to rise, and then activates the hinge and activates the B6, B9, and B3 to cause a very strong retraction, which leads to the peak force, which is maintained for a time as the animal attempts very hard to tear the seaweed, and then it relaxes the grasper, opens it, relaxes the jaws, and the force falls, but not all the way because the pinch is still present until the end of protraction. So that's the swallowing cycle for a large mechanical load. Finally, let's look at rejection. In rejection behavior, the animal is given something inedible, here a polyethylene tube, and after several attempts to swallow it, begins to recognize this is not food and rejects it. In this initial configuration, notice that there is a black ring on the polyethylene tube, and we've drawn a line to show how far out of the jaws it is when the animal begins the rejection behavior. We'll now run the movie of the rejection, and you can follow what's happening to that specific ring that's marked on the tube as the animal rejects the tube. And here we've drawn a line to indicate how far out it went compared to where it was originally. So you can see the animal is capable of significantly pushing materials out of its feeding apparatus. How does that work? Well, the first stage, and this is different from any of the ingestive behaviors, instead of coming out open and going back in closed, as you do in biting and swallowing, you start by closing because you're grasping the inedible material and pushing it out. And that's activating the B8A, B8B motor neurons, which you can see on the radial nerve in an intact bathing animal. Step 2, with the closed grasper, you use the I2 muscle to protract and push the inedible material out. And as you can see, I2 can be on for very powerfully for a fairly long time to ensure that the inedible material is pushed far out, as we saw in the previous video. Now, the next stage is that you need to retract with the grasper halves open. Now, you've stretched the hinge here, so you could activate the hinge, but it's very important that what you do is not close the lumen of the jaws, because since the lumen of the jaws surround the grasper, if that closes, it's going to push the grasper closed again. And we showed experimentally that in that case, you pull the food right back in. So you need some way of delaying the onset of activity in these motor neurons that would otherwise close the lumen. And you also want to ensure that the grasper is open. And so there's a multi-action neuron I mentioned, B4, B5, that inhibits all of these to ensure that that's going to happen. And then finally, if you want the retraction, then at the very end, you can activate the hinge and you can activate B6, B9, and B3. And that's what we see here. Actually, the hinge is activated early. Sorry, that's like in biting. That's where it plays a role in the initial retraction. Here comes the B4, B5 inhibiting activity in B6, B9, and B3, and in B8. You can see this is essentially off during the most intense firing here. And then there's as almost an afterthought, there's a little final retraction phase here. So we have gone through all the different behaviors. Let's summarize this by saying that what you can see is these are the 3 stages of biting, the 4 stages we went through in swallowing, and the 4 stages in rejection. I'll draw your attention to the fact that the I2, the protraction phase, always precedes activity in the retractor motor neurons, B6, B9. Here's I2 prior to B6, B9, B3, and here again is I2 prior to B6, B9, B3. But that the retraction process can be shared between B6, B9, and B7, depending on the context. That the timing of the B8, B8A and B8B motor neurons depends on whether you're dealing with biting, where it overlaps to help enhance the I2, where it almost does not at all overlap, so that the protraction is not being antagonized. You're not pushing the seaweed out by closing on it as you are trying to pull it back in. So there's a very little overlap here. And total overlap, in fact, starting earlier when you're trying to push inedible material out. I've also pointed out that you can bring in a new higher order multi-action neuron to actually ensure that certain components of the motor system are suppressed during the rejection phase when necessary, and that you can, we've also shown you can prolong activity in these depending on the strength of the load that the animal is dealing with. So all of these changes in phasing, timing, and duration allow a single animal using the same periphery and the same motor system to generate 3 qualitatively different behaviors. And this, I think, is not at all unique to Aplysia. I think this is something shared across the whole animal kingdom in terms of how motor systems can generate multiple forms of behavior. In a separate video, which will follow, I will talk in detail about the different references that are the basis for all the information that we're showing here. But again, I want to emphasize that these recordings that are shown here are all taken from the same animal and same intact behaving animal, and the same animal went through all of these different changes in its motor patterns as a function of what behavior it was doing, and we've seen this across many different animals. Thank you for your attention.